Fox is online. King is online. James is online. Green is online. Lewis is online. Honor is online. He is not here. Boggs is not here. Clay is not here. Dawson is not here. Ellis is here. Castro is not here. Harrison is here. Hinson is not here. Kesterson is not here.
Morrison's here.
That was almost perfect timing. Lindeque's not here. Morrison is here. Grace is not here. Ward is not here. Wilder is not here. And Parks is not here. And of course, if you're online, as usual, don't forget to send me the attendance words.
You're a fan of the Braves?
Yeah, I don't follow them or any sport like I used to, but I used to be a huge fan. I grew up, in Atlanta when I wasn't living in Florida. So yeah. How about you?
Unfortunately, I'm a Marlins fan.
Sorry.
Pretty rough.
Oh, let's talk about rough. I know how you feel.
Oh, you guys won a World Series.
Not this year though.
I mean, in the last 5 years,
Yeah.
the Marlins haven't been even close since 2003.
Now, I'd also liked Marlins when they first started because I was living in Key Largo at the time.
Yeah.
And I was still primarily a Braves fan, but I still liked the Marlins. Especially when they were the Florida Marlins and not the Miami Marlins now, right?
Yep. I went to 3 games when they were the Florida Marlins.
Yeah.
They still suck then. Those two.
Yeah. Well, when I like a team these days, it's always bad for them. I mean, I've never seen anything like that in Florida State. I mean, I've seen teams that bad before, but not after a year like last year.
They had a completely new quarterback.
Yeah.
And he has a track record of sucking.
Yeah. It's almost over. Aki is online. Let's see. One. All of this here. It's 8 o'clock. We'll get started. First word for attendance is gym, like the name gym, not the place that people work out. Name gym, J-I-M. Let's see, announcements, reminders. We are starting module 6. I guess we haven't started it. Anyway, the exam is open. Exam 3 is open, starting today, and then closes on Sunday. This is the last portion of it, so get it done whenever you want. It's as usual, it's online, 50 minutes to take it. If you need more than 50 minutes, contact me so we can meet online and I can see your screen and I can hear you and see you and ensure that you're taking a closed book exam. If I know you're taking a closed book exam, I don't care how long it takes you to take the exam. So just let me know. And remember, the quizzes are the study guides. They're meant for you to take them and take them as many times as you want. Remember, once you hit a high score, no matter what you score after that, that original high score is what stands. And for most of the quizzes, every time you take them, you should get a new set of questions. I mean, obviously, if you take it a lot, eventually you will see all the questions, but that's what you want, right? That's what's going to help you prepare for the exam. So any questions about the exam? This week's lab, again, it's online, it's virtual. You can come in if you want. And how do I put this? I highly recommend coming in though this time, because take a look at it between now and Wednesday and see if you think you can do it on your own without it being a big hassle. Because my plan is, for anybody who's here, I'm going to run the simulation and project it on the board right here, and we're going to do this together. And then I'll give you the data, right? Because we're going to kind of do that together, and we're going to talk about what the data means. You'll put the data into your tables, and I'll be here and I'll help you with the calculations. So again, you don't have to come in the lab this week to do that Hardy-Weinberg lab, but I recommend it. So what I really, really recommend is at least look at it between now and Wednesday to see if you think you're going to need some help. Other announcements to remind you: Friday, I will not be having office hours. Monday, we will not be meeting. So a week from today, we will not be meeting in person. Class will be online only. I think that's it. And independent work. Let's get some independent work done. And then finally, I'm sure you guys have seen the announcement online, but I am backed up as I usually am on Mondays. So if you need me in a hurry, meet me online instead of sending me an email, because it'll be a while before I get to your email. That's it. Does anybody have any questions about anything? Okay, let's go back to what we were talking about. So again, most of this chapter, the meat and potatoes of it was talking about this whole process of going from DNA to RNA to protein. So when you're studying, I would focus mostly on that. But yes, prior to discussing that stuff, we also talked about how we do DNA replication, right? So that was this first main bullet point. And now that we're past all that, we're going to talk some about mutations, and then we're going to talk about viruses and other non-cellular infectious agents. So this information, the mutation part, is very important as far as what we're doing next, right? Because after this, we're going to start learning about evolution. And you can't have evolution without mutation. So you guys should know that from the lab you just did. That peppered moth lab, that was the point, right? It wasn't smoke that caused that mutation. It was just a random mutation. You had this one light-colored moth who had a mutation in her reproductive cells, right? And then therefore, she put out a brood, a bunch of eggs of moths that were dark colored. And it just so happens that that place was covered in soot and nastiness. So those dark moths had an advantage, right? And because of that, they changed over time. But the point is, it all started with a mutation. So let's talk about mutations. And again, I'll remind you, for independent work, anything I teach you, I think this is a great paper. It's like, all right, direct quote. Here's exactly what the book said, right? And then, we're using quotation marks. And then a direct quote, like, also, here's exactly what this source says. And then you can use your own words. You can compare and contrast how what the book says is different than whatever source you find. But anyway, let's talk about mutations. The definition of a mutation is any change in the nucleotide sequence of the DNA. So those A's and T's and C's and G's, if you change one of those letters, that's what we call a mutation. It can involve large regions of chromosomes. So you might have like whole chunks of chromosomes missing or duplicated or put in the wrong way. We talked about that in the previous chapter. It can involve a single nucleotide there, and I'm going to give you some really good examples of that, and one of which we've already talked about, which is sickle cell. Or it could be anything in between, right? So we're going to talk about these things. As far as the exam is concerned, there's nothing too much important on this slide because it's more of an introduction to what we're about to talk about. But also, again, this is your official book definition of a mutation. Not that I'm going to ask you the definition. Most of the mutation questions you're going to get are, I'm going to say, all right, here's this type of mutation. I'm going to describe it, and then you have to tell me what kind of mutation it is based on the description. And we're about to get to that. But so again, for the exam, I'm not necessarily going to ask you to define what a mutation is. But this is worth looking up, though. Kesterson, who's not here right now, was talking about this last week. So this definition says it's any change in the nucleotide sequence of DNA, and that's what we're going to focus on for this entire section of the chapter. But think about it. There's also other mistakes that could be made, right? So what about the RNA? What if there's a mistake when the RNA polymerase is reading the DNA to make that mRNA, right? What if they screw that up? What if there's a mistake with the splicing, right? Because we know there's splicing. You get rid of the intron to keep the exons. So what if there's a mistake with that? What if there's a mistake when the tRNA comes in and reads its anticodon and says, okay, here's this amino acid, right? There's all kinds of mistakes that could be made that we don't even discuss. So you can look that up for anything at work if you want. Anyway, here we go. Here's some examples. Again, we're going to talk about this in more detail later, just because it's a really great example. But this is the mutation that causes sickle cell. So here's the original code, and obviously it's a huge, long gene that we're just focused in on this one codon. Remember, a codon is the 3-letter word. It's supposed to be CTT, which would then result in an mRNA that's the opposite, right? So GAA, and GAA codes for the particular amino acid GLU. So it sounds like a bunch of letters, and it is, and that's okay. The point is, this is what it's supposed to be, regardless of what those letters are. That just happens to be what they are. And this is what happens with this mutation. So instead of a T, we end up with an A. And therefore, instead of an A, we end up with a U. And therefore, instead of this GLU amino acid, we have this one other amino acid. So all it took was changing one single letter, and now you have this whole disease called sickle cell. And we're going to talk about it a little again later in this chapter. And also, next chapter, we talk about evolution. Because does anybody know which people on Earth, I guess you could say, have sickle cell more than other people per capita? It's people from not South African, but sub-Saharan African descent. And we'll talk about why. Because even though this is a mutation and it causes a disease, it also, in some cases, gives you an advantage. So we'll talk about that later. Anyway, let's get into the important stuff as far as the exam is concerned. There are 2 categories of mutations in a gene. There are nucleotide substitutions, and then there are nucleotide deletions or insertions. You definitely need to know that for the exam. I'm about to teach you how they're different, how they're the same. So those are the things you'll need to know. You'll need to be able to compare and contrast the difference between substitutions and deletions/insertions. And notice we're categorizing those 2 as the same, right? So we've got 1 bullet point for substitutions, and then we have 1 bullet point for deletions or insertions, right? I might as well just put a slash there, deletion/insertions, and you'll see why. Because as far as the effect it has on the process, deletions/insertions are very similar, but they're very different in substitutions. So let's talk about it. Are there any questions so far? All right, the one I showed you previously was a substitution, and here's another example of substitution. Here you can see what the code should be, right? And that code will lead to this particular strand of amino acids. But here we substituted 1 letter, so instead of a G, we have an A, and therefore instead of this GLY amino acid, we have this SER amino acid, all because we changed 1 letter. And remember, that might seem small. It's like, oh, you only changed 1 amino acid. What's the big deal? But like we just talked about with this real example of sickle cell, if you change this 1 amino acid, then you have this whole misshaped protein. And in that case, then you have this whole misshaped blood cell. And then in that case, you have this whole slew of symptoms because of the disease. So it can be a very big deal, these substitutions. Also, and I don't remember, I need to fix this PowerPoint, but I would also point out that sometimes, because I don't remember if this is on the PowerPoint or not, but might as well bring it up now. Sometimes you could do a substitution where you change one of these letters, but it still codes for the same amino acid, in which case everything's okay. I hope that makes sense, because remember, that wheel where I showed you how to do the translation, and you saw which codes said, bring me this amino acid, bring me that amino acid. A lot of times, there's more than 1 codon for 1 particular amino acid. So sometimes you can change one of these letters, but still have the same amino acid. And again, I don't remember if I have this in the PowerPoint or not, so that's why I wanted to bring it up now. But anyway, that is an example of a substitution. Any questions? Okay, and again, it could be bad. It seems small because you're only changing 1 amino acid, but it could be disastrous. But what could even be more disastrous, and this is why these other 2 are lumped together, is insertions/deletions. So here, this is an example of a deletion. Here you can see this is what it's supposed to be. So U, right, is what it's supposed to be. But in this case, we left out the U, or deleted it, so to speak. So that's been taken out of the genome. Now, the reason this one is so much worse than the other one is because not only have you changed this word, but you've changed everything downstream of it. And of course, mind you, we're just zoomed in. Remember, a gene would be very, 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 very long, and we're just zoomed into 1 small part of it. So you have to imagine, when you pull that out, now everything all the way that way is messed up, right? Because everything's shifted. So now the whole thing is going to be different, right? So that's even more disastrous. Anyway, that's an example of a deletion. Here's an example of an insertion. Again, same concept. Yeah, you changed one letter by accidentally inserting that one in there. But it wasn't just that one codon that you messed up. Now everything downstream is messed up. So in short, for the exam, you need to know that substitutions are not generally as bad as insertions/deletions. And we will actually come back to this concept again, but that's a good introduction. Any questions so far? All right, the next word for attendance will be mad. Like that, like you're angry, M-A-D, mad. This confused things a little bit more. So again, the three categories of deletions, or excuse me, substitution, my goodness. The three categories of mutations we've talked about are substitutions and then insertions/deletions. Now we're going to make it even more confusing. There's all these different ways to categorize them. Then we have something called a missense mutation. So, missense mutation also happens to be like the substitution that I showed you, right? So a missense mutation is any mutation that changes the amino acid coding. So far, every example I showed you was a missense. Because look, here we have, oops, hold on, let me get that. Here I get it. All right.
Can you say that in class, just listen?
I guess so, yeah. I mean, the official policy is no, but I don't care. But anyway, yes, so even though we were talking about insertions and deletions and not mutations, I can't think, because they're all mutations. Insertions, deletions, substitutions, right? Whichever ones we talked about, they all also happen to be what we call missense mutations, because again, we've changed the amino acid coding, right? So instead of having this GLY, we have the SCR. And of course, when we did the insertion and the deletion, we really were changing a lot of amino acids. So again, all those things I taught you, even though there were three separate categories, technically are all under the category of missense mutations. Then we have nonsense mutations, which I'm glad I talked about earlier. I just didn't give you the word. A nonsense mutation is when you have a mutation that changes the amino acid codons to a stop codon. We talked about that, I think it was on Friday. So again, a missense mutation changes the amino acid. A nonsense mutation is a premature stop codon. So it's like, remember what a stop codon does, it stops the process of translation, therefore the whole thing's over. Any questions between the difference of missense and nonsense mutation? All right, to make it even more complicated, and I don't do this, I didn't write this chapter. Well, deletions or insertions are also known as frame shift mutations. Because like I was telling you earlier, when you delete or insert that one, the one amino acid, the one nucleotide, then everything downstream of it is messed up. So here's another example of that. This one's not from your textbook, but it's a great example. So once again, this is the normal codons, right? CAT, TCA, whatever, and this is what the amino acids are supposed to be. But here, when you have a deletion or insertion, instead of reading this, and then this, and then this, so on and so forth, right? The whole thing just shifted by one. So instead of having a CAT, it now reads ATT. So everything downstream is now coding for a brand new or a different amino acid, right? And again, that's disastrous. So again, let me back up once again, because it's all these different categories. Talked about insertions, substitutions, and deletions. And all of those examples that I gave you also happen to be missense mutations because they changed the amino acid coding. Then, speaking of the deletions or insertions, those are also considered frame shift mutations because they've shifted the frame of the reading. And also they fall under the category of missense mutations. And finally, the nonsense mutations are the ones where you have a premature stop codon. So any questions about that? All right, here's a nice little table from the old textbook. So you don't have access to this. This is from the old textbook, so just download the PowerPoint if you want to study it. Here we have the different types of mutations, so substitution versus insertion/deletion, and the effects are missense, which we talked about, nonsense, which we talked about, frame shift, which we talked about, and then one more that I talked about earlier, but I haven't officially given it to you in writing, so here it is, a silent mutation. Remember I was giving you that example? I said sometimes you can change one letter in the codon, but it still codes for the same amino acid. So even though you changed the letter, you didn't change the polypeptide, you didn't change the protein. Therefore, that is a silent substitution, or excuse me, a silent mutation. So any questions about all these concepts? It's a lot to memorize. It's pretty simple, I think. It's just having to memorize it was the challenge. Yeah, I don't have any more about that. So any questions? All right. Mutations occur in a number of ways. I'll put a question mark on this because I'm not sure I'm going to ask you anything from this slide. But there could be errors in DNA replication or recombination. So when we talked about S phase of interphase, right? When we talked about DNA polymerase opening up the DNA double helix and reading the code and bringing in the new nucleotides, sometimes that's where the accidents happen. Then we also have physical and chemical agents called mutagens. The most common mutagens, at least the physical ones, are high energy radiation, like X-rays, UV light, right? This is why you're supposed to wear sunblock, or at least cover yourself, because UV light can mess up your DNA. It can give you mutations, which is how you get skin cancer, right? It's a mutation caused by the energy of the UV light, and that mutation leads to the stuff we talked about earlier when you learned the basics of cancer. Remember we talked about the cell cycle and how if you mess up one of those proteins that controls the cell cycle, then you have out of control growth. Well, that's a good example. Then there's chemical mutagens, and I don't think your textbook goes into that, so that's a good independent work topic. For 1 point, you could just send me an email and say, I looked it up, and according to this source, here's a list of chemical mutagens. Or if you want to, you could write a whole paper about it. There you go, there's an independent work topic for you. Chemical mutagens, which ones are there? I'm sure you could think of some, like for example, nicotine, right? Or the stuff that have on cigarettes. It's actually a lot of stuff that's bad for you in cigarettes. But anyway, you can look that up if you want.
About alcohol.
What's that?
Alcohol.
Yep.
Can you inherit from your parents? If they were alcoholic, can you become an alcoholic because they were?
I've read that, but I didn't read it in the context of a biology student or a biology professor, so I've never looked into it to confirm that, but yeah, I've heard that. I think that's one of those two things that's probably environmental and genetic, I bet. But yeah, and that's also a good thing to look up for independent work. And not just that, but anything you could think of, whether it be a good thing or a bad thing, ask yourself, is that genetic, environmental, or a mixture of both? Like musical abilities, for example. Both of my parents can play instruments, I can play instruments. Neither of my kids seem to be able to play instruments, so it's interesting. It makes me wonder, is that environmental? Probably not environmental, because both my parents weren't exactly good at playing music for me or with me. I just happen to know they play music, but I play music for my kids all the time. Like, play with them, and they just don't, they're not into it. My adopted daughter, on the other hand, she can sing and play good piano, but obviously that's not genetic. Anyway, again, great question, great independent work topic for anything. Is this a genetic thing or environmental or some mix? Athletic ability, music ability, alcoholism, anger issues, right? Good stuff, bad stuff. Think about anything you can think of. You could look it up for to see how that works. And probably a lot of the stuff you look up, you'll find we don't even know yet, which is fascinating and promising if you wanted to look into it for yourself, which you guys are biology majors, but you could always switch. Anyway, mutations are often harmful, but sometimes they can be beneficial, both in laboratories and in nature. Because again, without these mutations, we wouldn't have natural selection, right? So again, using those moths as an example, if they didn't mutate and have that one brood of black moths, then probably all those moths would have been killed, right? Because they were too light. But there just happened to be that mutation, and that happened to basically save that species in that area. So yeah, mutations are the source of the rich diversity of genes in the living world. Basically, that's where we get all our diversity from. So yeah, it's important. It's a great way to end the discussion about mutations because again, we'll pick it back up when we start talking about evolution in the next chapter. Because again, without this mutation, we don't have any variation, and without variation, you don't have evolution. Anyway, any questions about mutations? Because we are now officially done talking about mutations.
Was it a mutation that caused us to have blacks and whites different color of skins.
Yep.
It was a.
Because we were all, I mean, the scientific consensus is now that we were all blacker, dark. And then as we migrated out of Africa, some people in the Caucasus Mountains, that went into Europe, there was a mutation where they were lighter, and it actually ended up being beneficial for them to be lighter in that environment. So yeah, anything you can think of, well, not anything, but a lot of things you can think of where we have physical differences that are due to genes, that was because of a mutation. Blue eyes, that was a mutation.
Huh.
All kinds of stuff. It actually gets really, really fascinating and complicated. But we're just talking about basic mutations, and there's other things where some human tens of thousands of years ago had a virus that affected their DNA, and they were talking about big mutations. Anyway, yeah, if we have more time to talk about it, we would. I mean, that's a whole course. It's a whole course in evolution. We're just going to talk about it for one chapter. Anyway, any other questions? All right. Then let's talk about viruses and other non-cellular infectious or infection agents. Yeah. So this one's good. Again, we're just scratching the surface. This is a 100 level class for non-biology majors. So you're not going to be an expert at the end of this, but you're going to know the basics. And this will be important, especially these days. There's a lot of misinformation about vaccines and about viruses. So when you go to do your own research, I should put that in quotation marks, you'll not be starting from scratch, right? I'm going to give you the basic information. So let's talk about it. Viruses, they are not living. Technically, we don't consider them alive, but they do share characteristics of living organisms because they have genetic material in the form of nucleic acid that is packaged in highly organized structure. Just like anything that's alive, right? Even living things, we have genetic material in the form of nucleic acid that's packaged in highly organized structure. Excuse me. Of course, for us, it's chromosomes, right? So your textbook doesn't even talk about the highly organized structure. We'll talk about it a little bit as you're about to see, but we don't get too much into like the structure of their genetic material. We say it's either DNA or RNA, but that's it. So if you want to for independent work, you can dive into that as well. But anyway, again, viruses are not considered alive. Generally, some people still argue about this, but we generally don't consider them alive because, first of all, they're not cellular. And if you remember back to chapter 1 of the first things I taught you, that is the definition of life, or not the definition. Remember, that's different, not definition. I can't think today. It's a Monday. Anyway, the description of life, right? We had all those characteristics of life, and being cellular was one of them. And then finally, they can't reproduce on their own. So viruses cannot reproduce on their own. That's why they infect living things, because that's how that works. And we're about to talk more about how that works. But are there any questions so far? All right. So viruses are infectious particles consisting of genes in a box, basically. It's a great way to put it. Of course, the genes are different depending on the virus. The box is different depending on the virus, but generally speaking, they're all basically just genes in a box. They're all made up of nucleic acid wrapped up in a protein coat. So again, the type of nucleic acid depends on the virus. It could be DNA, it could be RNA. And of course, the protein coat itself might be different. And also sometimes there's things in addition to the protein coat. But at the very least, all viruses have a nucleic acid that is wrapped in a protein coat. And again, we'll talk about some of these differences, but they all at least have that. Yeah. Any questions so far? All right, and once again, and this is the second time you've seen this, so obviously it's important. Viruses cannot reproduce on their own. For that reason, they have to infect a living cell to direct its molecular machinery to make more viruses. And you already know, based off of what you just learned from earlier in the chapter, if it's a DNA virus, that means it needs your cell to make mRNA from the DNA, and then that mRNA has to be translated into a protein. And if it's an RNA virus, then it requires your cells to just translate that mRNA and make a protein. But here's a good visualization of what some of them look like. It's like the basic generic virus right there. Got genetic material on the inside. In this case, it just happened to be DNA instead of RNA, and then a protein coat on the outside. And again, like I said previously, sometimes there's also a membrane around it, just like our cells have a membrane. So some of these viruses are even closer to being alive or more like a cell, but not quite there. So let's talk about the different types. You're going to need to know the difference of these on the exam. Of course, it's all pretty much in the names. Bacteriophages, plant viruses, animal viruses, Then we'll talk about HIV, and then finally prions. All right. So, it's already up there. You guys already saw the answer. But if you didn't see it, if you had to guess, what do you think these types of viruses kill, or attack, or use, or infect, whatever word you want to use? Bacteriophages, what do you think they infect? It's in the name, bacteria. You need to know that for the exam. Viruses that attack bacteria are called bacteriophages, or phage. I've heard it pronounced both ways. Basically, that word just means bacteria eater. These things destroy bacteria, right? And sometimes they just call them phages or phages for short. And they consist of DNA enclosed in a structure of protein. So definitely know that bacteriophages attack bacteria, and maybe know that they're DNA, they are DNA viruses. And for independent work, you could look that up, because again, this is old. This is probably, 10 years old, this information? So I'm wondering if that's changed. Also, I'm wondering too, maybe if I just accidentally typed something, it's been that long since I thought about it, but look that up for independent work. Are all bacteriophages DNA viruses, or are some of them RNA viruses? Look that up if you want. And again, that could be a one-point thing where you just look it up, email me, say, according to this source, here's your answer. Or if you want to, you can use the Google Docs and write a whole paper about it and get your points based on how long your paper is. Here's what it looks like from a microscope. Right here at the bottom is a bacteria cell. You can see here that these things have attached themselves, and they've injected their DNA into the virus. And it's going to get a little bit more complicated. There's a drawing of what we're talking about. I think this is from the old textbook and not the new one. I don't remember. Doesn't matter. It's funny. They almost look like little syringes, right? And then, in a sense, they are. They attack the cell. They literally inject their genetic material into the bacteria. And the genetic material for this virus, just to make it easy to differentiate, they've painted it or colored it red. Meanwhile, they colored the genetic material for the bacteria green. And you can see what's happened here is the genetic material this DNA actually incorporated itself into the virus, into the bacterial chromosome. Remember, bacteria have one big circular chromosome. And in this case, they actually insert themselves into that chromosome. And actually, we'll talk a little bit about that when we talk about animal viruses, because some animal viruses do that to us. Some of our viruses that we get actually goes into our chromosomes, right? And it's there permanently. And then every time you go through mitosis, you copy, your whole genome, including that new virus. And again, going back to what I was saying earlier, we don't have time to talk about it, but some evolution stuff is really interesting that way. Like for humans, like we wouldn't be how we are if some ancestral human tens of thousands of years ago didn't get some virus that made huge changes to their genome. But anyway, let's talk a little bit more about this. Let's make it more complicated. So when we're talking about bacteriophages, there's two different types of cycles, and you need to know the difference between them. There's the lytic cycle and the lysogenic cycle. Before I even get into it, I'll tell you one of the things you need to remember is the lytic cycle is the shorter cycle, and the lysogenic cycle is the longer one. And when I was a freshman, the way I remembered that is because the word lytic is shorter than the word lysogenic. So I always remember the lytic cycle was the shorter one because it's a shorter name, and the lysogenic cycle was the longer one because it had the longer name. So let's talk about it. We'll talk about the lysogenic cycle first. This is where the viral DNA replication happens without a phage production or the death of the cell. So basically, this is what I was talking about earlier. In the lysogenic cycle, at first anyway, the DNA is inserted, but they don't start making the new viruses right away. Instead, that DNA gets inserted into the chromosome, like I mentioned earlier, and then it keeps getting duplicated and duplicated and duplicated without even turning into the new viruses, right? And there's a word for that, and that's another word you might need to know for the exam. I'm not going to ask you what this word means, but I might use that word in a question, and that word is prophage. So let me back up, and you can see this slide again. But this is what I'm talking about. When the DNA gets inserted, and then it inserts itself into the original bacterial chromosome, we now call that a prophage. That's what that is. And what you're seeing here, and I'm going to give you more pictures later, but here on the left, that's the lysogenic cycle, or the lytic cycle. That's the most basic, right? So in that case, on the left-hand side, the lytic cycle, the DNA is injected, right? And then the cellular machinery says, oh, that's the DNA. Let's transcribe it, and then let's translate it into proteins, and that's what happens, right? So the DNA that gets inserted is instructions on how to build more viruses. So it does. It reads the instructions, it builds more viruses, and eventually there's so many viruses in that cell, they explode, and then those viruses go out and infect other cells. That's the lytic cycle. The lysogenic cycle, again, is when the DNA is not used right away to make the proteins, to make the viruses, but instead it incorporates itself into the original chromosome, and then it just gets duplicated and duplicated. And it doesn't end in the death of a cell when it's in the lysogenic cell. The lysogenic cycle is not deadly until something kicks it on, and then it goes into the lytic cycle. And then that DNA is read, and those instructions on how to make the viruses are followed, and the viruses are made, and again, that is the lytic cycle. If you download this PowerPoint, you can watch an animation that shows this. Same here, here's another animation that'll show what I'm talking about, and here's a great little picture. This is not from your textbook, this is from the old textbook. So, if you like this picture better than the one in your textbook, you're gonna need to download the PowerPoint to study it. But this is the lytic cycle, right? So this is the drawing of what we talked about. Once again, you have this virus, it inserts its DNA into the bacteria, right? In this case, it doesn't incorporate itself into the bacterial chromosome, it's just sitting there. And then the instructions on that DNA on how to build new proteins, specifically viruses, those instructions are read. We make more viruses. Eventually, there's so many viruses that the cell explodes, and then those viruses go on to infect another cell, and the cycle continues. And that is the lytic cycle. So again, that's the lytic cycle. Are there any questions about the lytic cycle? All right. Again, lysogenic cycle. Like I said earlier, the DNA is inserted, but at this time, instead of that DNA, those instructions on how to build the viruses, this time they're not read. This time, that DNA is incorporated into the original chromosome, and then every time this thing goes through mitosis, its whole chromosome is being duplicated, including this new viral DNA. And that's what happens. And eventually you just have millions of cells that have this new virus in it, and they're just not doing anything yet as far as the virus is concerned. But then again, and this picture puts them together, eventually these cells that were in the lysogenic cycle do go into the lytic cycle. So remember, lysogenic is longer, lytic is shorter. Lysogenic cycle doesn't kill the bacteria, the lytic cycle does. Now again, generally speaking, the lysogenic cycle will lead to the lytic cycle, but until it does, it's not deadly. Any questions about those viruses? All right, here's the easiest question of the day. And for this one, I'm really not going to move forward until someone answers it. It's this easy. What kind of organisms do you think plant viruses attack? So we just got to finish talking about bacteriophages, which attack bacteria. What type of organisms do you think plant viruses attack?
Plants.
Plants, thank you. Yes, it's very easy, right? Hopefully that was just so easy. We're just thinking, is this a trick question? But no, it's not a trick question. What's interesting to me about plant viruses is they're generally not necessarily deadly, but they're not good for the plants. So they stop plant growth, they diminish crop yields, which is what's important for us, right? We need crops. But yeah, they're just generally not deadly. You should also know this. Generally speaking, plant viruses are usually RNA viruses, which again would be another great independent work topic. What are the exceptions? Like I said, generally they are RNA viruses, but which plant viruses are DNA viruses? Also, is there a reason? Why are most plant viruses RNA? What's wrong with DNA viruses? Your book does give an example, both the old textbook and the new textbook, and they talk about tobacco mosaic virus, which we abbreviate as TMV. I'm not going to ask you any questions about this, but we will talk about them very quickly. TMVs are rod-shaped, as you can see up at the top right. That's a good picture of one, with a spiral arrangement of proteins. It's a little bit hard to see there, but each one of those little purple things is a protein, and it's just kind of spiraling around it. And it affects tobaccos and related plants. Again, good independent work topic. I just said it affects tobacco and related plants. So what are the related plants? Not that you need to know, but again, for independent work, you can look it up. What it does, it causes discolored spots on the leaves. And I think the reason why both this textbook and the older textbook talk about it, not because it's just really cool or anything. It just happens to be the first virus that was ever discovered. Also, another good independent topic. When was it discovered? How was it discovered? Who discovered it? Did that lead to anything else? You know, you can look into that. Anyway, here's some pictures of what we're talking about. I don't know if you've ever seen a healthy tobacco leaf, but they don't look like that. This is a tobacco leaf that's been infected by this TMV. Here's a picture of it, right? There's the entire virus. If you zoom into this one section of the virus, you can see these proteins spiraling around it, but also RNA that is spiraled inside. Anyway, any questions about TMV? We're going to talk a little bit more about plant viruses. All right. Plants don't necessarily have a respiration system. They have stomata, where they let gas in and out, but they don't necessarily have a respiration system. So how do the viruses get into the plants? Well, they have to get past the plant epidermis. So usually it's a damaged plant that's more likely to be infected, right? So this is why in farming, they're doing it right. They're very careful with their tools. If you cut one plant that has the virus, now you've got that virus on your tool, and if you cut another plant, then you've infected it, right? There are no cures for most plant virus diseases. Another good independent work topic. Why not? What is it about these plant viruses that make them so hard to cure? Also, I just use the word most, right? So what are the exceptions to that rule? But because there is no cure for most viral plant diseases, agricultural scientists usually focus on preventing the diseases to begin with and breeding or genetically engineering varieties of these plants that can't be infected. Anyway, that's it for plant viruses. Any questions about plant viruses?
Poison ivy, a plant virus?
Nope, good question. No. Poison ivy is just a plant, that has oils that cause reactions on humans. So plant viruses are the viruses that attack plants, as opposed to plants that attack humans. But you know what? Now I'm curious, and I know it's not a virus, but I'm curious, are there specific viruses that do attack poison ivy that don't attack other things? Something you could look into for independent work. Any other questions about plant viruses? All right. Next thing we're going to talk about is animal viruses, which brings up the next independent word, the next attendance word, which will be dog. I know none of you are biologists, but a dog is a type of animal. Now you know. You can write home and tell your parents you learned something in college. Anyway, animal viruses are very common causes of disease, as you're very aware of, especially now. We're just, COVID-19 is still out there, right? But we just went through a pandemic of it. So you guys are very familiar with animal viruses. This book was written before COVID-19 came on the scene. So back then, that was correct. And right now, I'm not sure if this is true or not. I know for sure for when COVID-19 first hit, it was deadlier than flu. But now I don't know if that's the case anymore. Because, first of all, we've learned how to prevent it. We've learned a little bit how to treat it. And sadly, I hate to say this, but the people who are more susceptible to dying from it did so, right? So unfortunately, it's an example of natural selection. A lot of us who are here, how many of you don't have to answer this because this is HIPAA, but if you want to, how many of you have had COVID? Because I've had it a couple of times. So I know for a fact there's something in me genetically that means it doesn't kill me. Meanwhile, there's something genetically in other people, amongst other things, not all genes, but it kills them. Anyway, that out of the way, let's pretend like COVID didn't exist and talk about this like I was giving this lecture in 2018, right? So for the whole human history, for sure, flu was actually has been the worst. And we don't think about it much because, again, like those of us who have survived COVID, people get the flu all the time. And I know I never thought about it until I was in biology. Like, I had the flu, I survived. I know people who had the flu, they survived. But it actually has been devastating for humans in history. Anyway, like many animal viruses, flu has an outer envelope with a phospholipid membrane that has spikes of protein. So you might want to know that. I might ask you that. Not as specifically that flu has an envelope of phospholipid membrane, but what I'm saying here that you need to know possibly is that many animal viruses have a phospholipid membrane, which is what we have, right? Which makes sense because that's how they kind of get into our cells, because they also have a membrane, just like ours. Of course, theirs have these spikes of protein, which, just so you know, that's basically how that mRNA vaccine worked for COVID. So there's one of those spikes, and the spikes themselves are harmless, but they're specific to that virus. So the people who were in charge of it, they figured out the genetic code to make that protein, and they give you the mRNA for it, and your cell is like, oh, I'm going to follow these directions, and they make that protein, and your body knows to attack it, right? So it basically trained your body to attack anything with that protein, which is the COVID-19 virus, without giving you the virus itself, just the protein. But anyway, once again, for the exam, the important thing here is that you need to know that many animal viruses have a phospholipid membrane. Anyway, any questions so far? All right, so here's some examples. Again, they could be RNA, they could be DNA. Here's some examples of both. RNA viruses include flu, common cold, which is what COVID-19 came from, basically. That's COVID is a common cold. Measles, mumps, AIDS, polio, and then some DNA examples include hepatitis, chickenpox, and herpes. And we're going to talk about all of those individually. I will put a big question mark to this. I'll put 2 question marks. Like, I'm really doubting it. I really, really don't think I'm going to ask you to differentiate. Like, I don't think I'm going to give you this list and like, which one of these is RNA viruses versus DNA viruses? For some of these, you'll probably remember once we talk about them and talk about how they work, but probably I'm not going to ask you that. Any questions about that slide? All right. Here's a picture of one. This should look very familiar, right? This looks a little bit like the plant virus I just showed you, that TMV. It's a spiral of RNA coated in a spiral of protein, except in this case, that is also encased in a membrane, right? So again, so we can get into the cell membrane of the animal. Because remember, plant cells have a cell wall. So it's not like you could even use the membrane to get into the cell wall or to get into the cell, right? Because they would have to get through the cell wall of the plant. Animals, we don't have cell walls, right? So you can just kind of merge right into the cell membrane, just like this picture shows. So here's how it works. Again, it's very similar to what I showed you earlier. The concepts are basically the same for a 100 level non-biology major class. Just like last time, the genetic material enters the cell. In this case, they get in even more detail and talk about how that protein code is taken away so that we can get to the genetic material. Again, that genetic material is then synthesized, or we have an enzyme, a virus enzyme that kind of duplicates it. I'm not even going to get that duplicated into this for the exam. And then some of that is used, right? So again, we've duplicated that genetic material. Some of it is then used, right? It's packaged up in the protein and put into the virus. Actually, both of those, that's what's happening. This was used as protein synthesis, right? So that's the code that says, all right, build this protein, build this protein, build this protein. That was used for that. This one was like, all right, well, we need to build more viruses, and those viruses need to include genetic material, so let's duplicate the genetic material so we can put it into the virus, which should make sense. So again, big picture here. Once that genetic material enters the cell, like we see here, half of it is used for the code to build the new proteins, and then half of it is used to make the new proteins. And then again, just like I showed you with the bacteria phase, it reproduces, reproduces, reproduces. Eventually, it reproduces so much it basically blows the cell up, and then the cells leave. The virus leaves the cell, and then it's able to go on and infect another cell. And here's a picture that puts it all together. And that picture is from the old textbook. So again, if that picture makes sense to you, more than the one in your textbook, and you want to study it, you'll need to download the PowerPoint. Yep, but that's it in a nutshell. Any questions about that? All right. Here we have an animation you can watch that shows how an animal virus works. There's a simplified reproductive cycle. Here's a picture, again, from the old textbook. But that's an animal cell being infected by the mumps virus. So you can see, obviously, the big thing in the middle is the animal cell. These little things on the surface of the cell are the mumps virus. Excuse me, I'm looking at this the wrong way. The whole thing is the virus. I'm sorry. My coffee hasn't kicked in yet. This whole thing is the mumps virus. And these little things sticking out of it are the protein spikes. And again, also, like this points out, you can also see the envelope, right? That phospholipid bilayer, just like we have in our cells. So, any questions about that? All right. Not all viruses reproduce in the cytoplasm. So the one I just showed you that drawing where it was inserted and then unraveled and then duplicated and then repackaged, right? That was happening in the cytoplasm, but not all of them do. So you might need to know this for the exam. Herpes viruses, which include chickenpox, shingles, cold sores, and genital herpes, do not reproduce in the cytoplasm. They are enveloped DNA viruses, and they reproduce in the cell's nucleus. And they get their envelopes from the cell's nuclear membrane. So I'm not going to get that deep for sure on the exam. I'm not going to have you memorize all that. But you might need to know that the herpes virus is one of those that's a DNA virus, right? It happens inside the nucleus, not inside the cytoplasm. We're going to talk a little bit more about it, so I won't ask you if you have any questions yet. But again, the herpes virus DNA remains behind in the nuclei of certain nerve cells. And this is why, I don't know if you've ever heard the term, the herpes virus is the gift that keeps on giving. That's why, because it's not like the genetic material just goes in your cells and your cells read it and build a new virus. Instead, it gets incorporated into your genome, right? Very often it remains dormant until stress kicks it off, like if you have an illness or emotional stress, something like that, that will kick off whatever, the DNA itself, then you'll have shingles or whatever it might be. Usually it's very unpleasant symptoms, and it may flare up repeatedly throughout your life. And then your book also points out that over 75% of adults have herpes simplex 1, which is cold sores, and about 20% have herpes simplex 2, which is genital herpes. Also, and I don't remember if your textbook gets into this, but basically, shingles comes from chickenpox. So if you got chickenpox, it's in your genome, and then again, it usually remains dormant until something kicks it off, and then you get shingles.
I've had shingles and it hurts.
Yeah, I had it too, which is not really normal because I was in my 20s, and this usually people that young don't get it. I was in a very stressful situation, so it kicked off, and that certainly didn't help things. But anyway, so as far as animal viruses are concerned, some of them are worse than others, as you already know, and the damage depends partly on how quickly the immune system can respond to the viral infection, but also the ability of the tissue to repair itself. Because remember, not all viruses attack the same thing, right? Some of them attack your respiratory system, some of them attack your nervous system, right? This is why people very often fully recover from colds, because the respiratory tract is actually very efficient at replacing its damaged cells. That's one of the reasons. And also, not everybody fully recovers from colds, which is kind of the opposite of poliovirus, for example. This is just one example, but poliovirus, which is almost eradicated, starting to make a comeback now. Anyway, it attacks the nerve cells. The nerve cells are not usually replaceable, so that's permanent damage, right? That's why people used to have the iron lung, because they literally just couldn't breathe, so they had to have something, breathing for them, basically. Anyway, any questions about that? If you download this PowerPoint, you can read this little section called The Process of Science. Again, this is adopted from the old textbook. I kept it in the PowerPoints just in case you guys want to read about it, but I never will test you on it. And the last word for attendance is going to be Mattis. Mattis, like the last name. Mattis. That's it. And when we come back on Wednesday, we will finish this chapter and start the new one. But remember, this chapter is the last chapter that's going to be on this exam. So I'll be online if you guys need me for office hours. And don't forget to take a look at that lab and see if you think you should come in on Wednesday, because it really is up to you. And if you think it's hard and need help, then you should come in.
I came in probably 1 to 2 minutes late.
Yeah, if you don't mind, just send me an email to remind me.
And then I was going to see, are you still opening chapter 5 test? Or am I too late to do? It was just last week. It was due the 27th, I think.
The Exam 2?
Quiz 5.
Oh, quiz 5. Yeah, I can do that. Send me an email again. That could be the same email. So when you email, remind me of that too. Yeah.
What's our subject today? Is?
Late, quiz 5, something like that.